So good afternoon, everyone, and thank you for coming. My name is Leslie, and my partner here is Anthony. We actually have 3 other group members that are holding down the fort at our poster. They're Alex, Pawel, and Sonya. And we're here to talk to you today about our Evergrow fertilizer, which is a new product that has allowed gardening to be made simple. And everything about Evergrow, from synthesis to application, is simple and safe, and we're going to walk you through that full process today. So quickly, an outline of what we're going to talk about in the next 20 minutes. I will introduce you to the problem, the current fertilizers on the market, and the problems they pose. I will propose our solution, the Evergrow fertilizer. I will walk through the design, synthesis, and characterization of what we did to characterize our Evergrow particle. I'll go through the full-scale plant study we completed to compare our Evergrow fertilizer to 3 other series of plants. And finally, we'll talk about the achievements and customer requirements that were met, in addition to conclusions and future work that we hope to do. So first of all, plants need a wide variety of nutrients, but there's 3 macronutrients that they need more so than anything else. These are nitrogen, phosphorus, and potassium, or NPK. And the conventional fertilizer model that's used right now is a burst release model. So these nutrients are delivered to the plant, and a lot of the nutrients are run off, and they get into our rivers and lakes, as shown in the top photo there. These can cause algae blooms and other negative environmental impacts. This method also has a lot of fertilizer burn. So the bottom photo here, you'll see brown leaves on the side of the plant, and they start to curl in, and eventually this will kill the plant if it's given too many nutrients too often. They also require frequent reapplication, which is time-consuming and inconvenient. So there is an ever-growing market for fertilizer in Canada. In 2013 to 14, we see a 2.4% increase in the global fertilizer market demand. We have 2 main avenues in Canada where we see fertilizer. First, we see it agricultural and farms. It rose to $4.8 billion in 2012. And similarly, we have a home gardening market, almost as big as the farm fertilizer. It's expected to reach $4.2 billion by 2017. So here are 3 slow-release fertilizers that are currently on the market. So they would be our competition, if you will. All 3 of these use a polymeric membrane around their fertilizer nutrients, which requires to be broken down before the nutrients can get to the plant. The problem with this is that to break that down, it's very temperature dependent, soil dependent, and weather dependent. So in a climate like Canada, where we don't have a lot of consistency, it's not guaranteed that these nutrients can get to the plant. Alternatively, our Evergrow solution doesn't depend on these things. It's not temperature or soil dependent, so you're guaranteed to have nutrients being delivered to your plant. It also has an indefinite shelf life, and it's degradable and completely biocompatible. Admittedly, right now, because it's in the research and development phase, it is a little bit more expensive than the competition that's on the market, but we're hoping to lower these costs in future work.
So the product we're presenting to you today is Evergrow. Oh yeah, I gotta take this. Excuse me. Okay. So this is what our particle looks like. As you can see, there's a mesoporous silica core that is loaded with fertilizer, and this is all encapsulated in a water retention layer, which is sodium alginate, and together they provide the slow release capabilities that we're looking for. So our product gives you low maintenance. That is, you apply the fertilizer once, and it is able to deliver the fertilizer payload over a longer period of time, as opposed to traditional fertilizers, and it is also environmentally safe. This is a brief look of what it looks like, but I'll go into it later. So the detailed design of a fertilizer, we have the mesoporous silica cores. These have a dense pore network inside, and using these pores, we're able to load the fertilizer into these pores. And because of the tortuosity of the pores, the out-diffusion of fertilizer is delayed, and thus we get controlled release of fertilizer. The outside, the sodium alginate layer, is able to impart some structural support to the mesoporous silica cores, and also it's able to capture and hold, and then subsequently release the water and fertilizer components. So before we set out to synthesize this, we tried to simulate what a particle should have as its properties. Using literature values, we got the ideal particle, and we analyzed for how it should behave in a situation such as in a plant. However, it is very difficult to simulate the dense pore networks inside the mesoporous silica, and without doing molecular modeling, we can only we could only simplify the solution down into simple diffusion, and that we feel is perhaps not completely accurate. However, using this simulation, we were able to get some realistic expectations of what we should see in our particle, and we felt confident enough to carry on to synthesis. So the synthesis of the particle is conducted in 3 simple steps. First, we generated the mesoporous silica pore, and then we load the fertilizer in it. Subsequently, we encapsulate it in the sodium alginate layer, as I mentioned before. To generate the mesoporous silica core, we use sol-gel templating of TiOS and tergital. So the TiOS is the silica source, and the tergital is a non-ionic surfactant. We mix that, we let that sit overnight to self-assemble. To tune the pore size of the final mesoporous silica, we can change the pH, the amount of sodium fluoride, and also the temperature. So the next day, we add sodium fluoride to catalyze the condensation. As you can see here, you start to see cloudiness. This is the silica particles inside the solution. Then we filter it out, and then we calcine it to burn all the organic parts of the starting solution away.
So once the silica core has been created, we have to load the fertilizer into those pores. So this was done by dissolving fertilizer in water and adding the silica and putting it in a vacuum environment where the fertilizer is essentially forced to go into the pores of the silica. And then once dried and the water's evaporated, you're left with just silica with fertilizer inside the pores. I think we have a picture here too. You can see it. Well, it's slightly blue in color. You'll see them across the bottom. So once it's loaded into the silica core, we have to encapsulate it in an alginate layer. So this was done by taking the loaded silica and dispersing it in the alginate. The alginate is then taken and dropped into a calcium chloride solution, and reverse spherification happens, and you'll see beads formed. We actually have a couple of pictures and a video to show you this process. So here you'll see the loaded silica, the powder on top, being stirred and dispersed in the alginate solution. And here's a video showing the beads forming. So that blue vial that's being held up is the silica dispersed in the alginate. And then using a pipette dropper, we can take that alginate and drop it into the calcium chloride. The calcium ions interact with the alginate and gelification happens. So we see reverse spherification and beads start to form. So you'll see that in a second. And depending on the size of the pipette tip we use and the concentration of alginate, we can control the size of the beads formed. So you'll see when the alginate hits the surface of the calcium chloride solution, it immediately forms the bead and it falls to the bottom of the beaker.
It's a little hard to see, but you see these little blue beads flowing around.
So here's the final product once all the beads have been dropped. What's important here is that the beads, the fertilizer was contained in the beads. It didn't leak out into the calcium chloride solution. The solution is clear as when it started.
So in order to make sure that the fertilizer particle we make is able to perform to its design properties, we set out to characterize every part of the synthesis process. So we started with the mesoporous silica core. We tested the amount of surface area it has and the pore area. And also we set out to determine how much fertilizer we can load into this silica particle. Then we went on to characterize the water retention properties of the alginate, since we said that it should be able to retain some water. And then, satisfied with the results, we went on to see how the final particle looks and whether or not it conforms with, say, for example, current fertilizers on the market. Lastly, we went on to a lab scale study and to show that our fertilizer, once encapsulated, is still able to be released. So first, this is an SEM image of the mesoporous silica cores. So this is before loading. The idea of this is to show that even after loading it with fertilizer, that the morphology stays the same and that nothing has changed, and also that the fertilizer is actually being loaded into the pores and not just crystallized around these particles, for example. So here you can see that the average particle diameter is around 1 to 4 microns, which is actually in our benefit because a different sized particle will provide a different release profile, and hence we should get a total longer release. So this is the particles that, after they've been loaded with the fertilizer. Here we see that the morphology of the particles have not changed, and thus we can conclude that all of the fertilizer has been loaded into the pores of the silica. So next, we set out to figure out or get an idea of how much fertilizer we should be able to load into the silica. So we did nitrogen absorption measurements, and we figured out that the surface area was at least 1,000 meters squared per gram, and also that the pore radius was around 1.5 nanometers. This satisfies both our customer requirements for surface area and radius. Next, we ran a series of tests to determine exactly how much fertilizer we can load. And as you can see here, we have a series of concentrations of fertilizer, and then we set out to impregnate these into the silica. And we determined that we can load about 2 grams of fertilizer in a gram of silica. And lastly, the alginate layer. So we measured the weight of these alginate beads when they were formed and compared it to their weight after they've been completely dried. And we found that they were able to hold 117 times their own weight in water. And then, after they've been dried, so this is what they look like when they're dry, we resubjected them to water and determined that they're able to reabsorb 11 times their own weight in water. So as a final Evergrow particle size, using the different techniques as Leslie has talked about, we can fine-tune the size. Currently, using our techniques, we can tune the size between 3.5 to 5 millimeters in diameter. This is quite an ideal size because it's also similar to the size of conventional slow-release fertilizers. So it will be applied using the same process, and people are used to seeing fertilizer at this size, etc. And finally, in the lab study, to ensure that we can deliver our promises, we characterize the individual fertilizer components as they are being released from the particle throughout a 2-week period. And we found that nitrogen and potassium were being released at a rate that we expect. Interesting to note that phosphorus, while it seems low here, the reason for that is actually because of a limitation in the quantification method we have for quantifying phosphorus levels in the fertilizer. The assay we're using right now is only able to quantify phosphate. However, in a fertilizer, the phosphorus is present in other species as well, such as P2O5. And therefore, while this trace does seem low, this doesn't represent the total amount of phosphorus that we have in our fertilizer. That we should expect should be closer to the level of nitrogen and potassium.
All right, so once we were confident that the Evergrow fertilizer would deliver the nutrients to the plant as required, we went on to do our plant study. So the Evergrow fertilizer here is shown on the right, and we actually brought 4 of our plants from the study. We compared it to 3 other series of plants. So first we had a control. The plant only received distilled water. It didn't receive nutrients or fertilizer in any type. Second, we used the competition, the standard slow-release fertilizer that's on the market today. We also compared it to standard commercial NPK 20-20-20 fertilizer. So this study was done over a 1-month growth period, and 36 plants were used in total, so 9 plants of each series. So to control the external conditions of our study, we used the greenhouse here on campus. This gave us control over the temperature and humidity conditions. We also grew all the plants in turfus instead of soil. And every plant was given 250 milliliters of distilled water every other day, including weekends. And the greenhouse allowed us to use artificial light to consistently give the plants 16-hour photo periods. So once done, we used 4 measurements to quantify the results. These include stem circumference from the base of the stem where it sprouts out of the turfus, the height, the weight of the roots, and the weight of the stem and leaves. So before I go into statistical analysis, we have two pretty cool photos here to show you. So this is day 1. The Evergrow fertilizer here is shown on the second from the right. You'll see here the plants have just sprouted. They've just germinated. We can't give the plants fertilizer until they've sprouted. The seeds would die under fertilizer conditions. And then after a 1-month growth period, you'll see here that the Evergrow is towering over the other ones. I want to draw your attention specifically to the far left-hand side, the slow release. That's the competition. That's what's currently on the market today for a slow release fertilizer. This photo here just kind of gives you a better representation of the height of the plants once they've been harvested and uprooted. Give you an idea for scale. The Evergrow plant across the bottom is about just over 1 meter in length. And in addition to the height for the Evergrow plant, we also saw that it was better in all other measurements taken. So it's kind of faint here, but you can see that the Evergrow is green. It was far superior to the other slow release fertilizer that we used, shown in the yellow. So again, we did statistical analysis, and with a high level of confidence, we can conclude that the Evergrow fertilizer was far better than the standard commercial fertilizer, slow release fertilizer on the market today, and the water control. And we can conclude that it was statistically equivalent to the standard NPK fertilizer. However, here, it's important to note that the Evergrow fertilizer was only given to the plant one time. After one application, only water was given to the plant, and the plant grew equivalently to the NPK 20-20-20 standard. For this plant, you have to take the correct amount of fertilizer, dissolve it in the correct amount of water, and apply it to the plant every time you water it. It's time-consuming, and it's hard and difficult to do.
So to quickly recap on our customer requirements and the achievements we've shown, we were able to meet all the primary customer requirements. We formulated a fertilizer pellet within 5 to 15 millimeters. We were able to encapsulate the NPK fertilizer, and the alginate layer outside of the particles was able to absorb more than 100 times its own weight. Furthermore, we also met all our secondary customer requirements in terms of the mesoporous silica pore diameter, as well as the internal surface area. And as Leslie has mentioned, we were able to show controlled release of fertilizer over the 1-month growth period. In conclusion, our product Evergrow simplifies greatly the gardening experience. It allows you to have a one-time application and a sustained release for a longer period of time as opposed to conventional fertilizers. It is all green, and it shows statistically proven plant growth compared to the others. You can also see that here. And we were able to meet all primary and secondary customer requirements. So as future work, we'd like to improve on the structural integrity of our particles, for example, by integrating additives. And we would like to further refine the measurement of the release profile, particularly for phosphorus, to get a better idea of how exactly it is being released. And lastly, because of the research scale of our current work, the costs are a bit higher than competitors. However, we do expect that if we were to scale this up, that the cost would drop as well. So we want to thank you for your time for today, and we'll open up the floor for questions. I guess I'll keep this.
You just hold it.
Okay.
Questions? So you gelled it, right, to form those beads?
Yeah.
Okay, so the beads is actually containing water,
Sorry?
The alginate beads.
the algae beads.
Oh, yes.
It's just got like jello kind of things, right?
I can't find the mouse cursor. So we have the mesoporous silica particles.
In the solution, and you add the basically polymerize and form the gel beads, right?
Yeah.
So the beads itself contain water.
So yeah, we did do tests of that. However, we found that when the inside of the alginate is just water, that the nutrients quickly or quicker came out of the silica and into just the alginate layer. And since the alginate layer has a higher diffusivity, the nutrients also got out faster. So we also did a series of tests in trying different levels of fertilizer also within the alginate layer. So since if we have a higher concentration of alginate fertilizer in the alginate layer, the out diffusion of fertilizer components from within the silica pore would also be delayed.
Yeah, so what I meant is like the gel, it's basically a gel, right?
Yes.
So how do you put the fertilizer, just cover the top? Or you mix it with the soil?
No,
Oh.
it just covers the top. So
Yeah.
once the plant is germinated, once the seed is sprouted, you can put the fertilizer just on top, and when you water it, the
Sure.
fertilizer will seep down into the plant.
Okay, here's a question. You basically putting something really moist on top of the soil, like that. So basically, you are artificially, go back to the next one, previous one.
Oh, yes.
You are actually preferentially making your Evergrow soil to be constantly moist. Because when you compare with the other three cases, you have nothing on top. So the moisture can evaporate much faster than the Evergrow.
So these Evergrow particles, they look, because this is right when they were first applied, they do dry up, and then when we water them, they swell a bit, but it doesn't cover the surface like that for the
You
entire
can
study.
see here.
You can see, yeah, it's kind of dried on top there on the surface.
So the full coverage is usually on top.
You can put it across the entire top, you just need to leave a base around the stem of the plant. You
Yeah.
can't change the height from where the plant sprouted, or it'll kill the
Yeah.
plant. But it'll dry up and it won't cover the full surface, it doesn't need to cover the full
I'm
surface.
just kind of curious your experiment. I mean, is the gel preparation makes any difference? Because let's say if the other conditions, you didn't put the nanoparticles, didn't put the silica particles in it, just put the gel on top. Are you going to make the other plants grow faster too?
That would be an interesting thing to test. However, I can tell you that this substrate that we're growing the plants in, it's turfus, it's calcined clay, it has absolutely no nutrients, and because of that, we can show that all of the nutrients delivered to the plants are from whatever we put in them.
Water.
Yes.
When you put the gel on top, the gel itself means absorb moisture.
Yes,
So you
okay.
cover the top. So actually it can help slow down the moisture being evaporated from the soil.
Okay.
So if you put a 20-20-20 with gels only, without fertilizer, your beads only without the silica particle, is it going to make 20-20 make better?
It could make it, it definitely could make it better, but again, there you go back to the fertilizer runoff. So when you water that plant with 300 parts per million, we see that 300 parts per million run off. So we see a lot of excess nutrients being run off into it, whereas the Evergrow one gives the plant the nutrients slowly, so we don't see as much fertilizer runoff and the subsequent environmental impacts.
And the particles here shown, so with that water absorption ratio test, the one where they dried up and I compared the weights, they actually dried up within a day in the greenhouse environment because it's a lot warmer. So after they dried up, they look like this on this plant here. So I think there's minimal coverage after 1 day.
Okay.
Another thing is when you prepare the gel, there's a lot of calcium, right? So is that also a nutrient or?
Yeah, that's a good question. Actually, in the NPK fertilizer, there is sources of calcium. Yeah, so.
So it's more important for this study because the turfus has no other nutrients in it. Most people don't grow plants in turfus. It's not a good medium to do it. It's just good for studies like this where we can prove that the fertilizer is actually doing what it's supposed to do. But the plants need such a small amount of other nutrients that the soil usually has those in it. So the big ones are nitrogen, phosphorus, and potassium, but you can buy different fertilizers and we can load it with different things depending on the type of plant and the plant requirements.
The blue stuff, your fertilizer, right?
Yeah, the standard NPK comes in a blue color.
And that's like it's a single compound or it's like a mixture of many stuff?
So 20-20-20 stands for weight percent.
Right.
So 20 weight percent nitrogen source, P sources, and K sources. The rest are actually mainly filler. There's really little trace elements of, for example, magnesium. There's some copper in there, but these are usually 0.02% or something like that.
Okay. Awesome.
Yeah, I don't know. I mean, I just love plants myself. So this is
So do
very
we.
interesting. So I remember you've seen before. Some people actually put some sponge right on top of the plant.
Yeah.
So in itself, they are doing exactly what you're doing already. That because when you have a sponge always sitting on top, when you put the nutrients in it, it's always the sponge itself absorbs certain things. So the next time when you put just water, it slowly get the slow delivery of the I mean, I was just kind of wondering,
Yeah.
is it a cheaper way to do exactly the same thing?
Again, well, we're hoping to lower our costs when we do the scale up,
Yeah.
but for, I guess, like farm applications or anything else, to take a sponge and put it in each individual plant would be quite a lot of work. And depending on the size of the plant, having a sponge,
Right.
you're going to have nutrients delivered to one specific part of that plant. It's not going to reach the full level of roots that are there. Where if you have this fertilizer kind of dispersed on the surface, you're going to get a more accurate fertilization.
That's the problem, why the farm put wheat straw, right, underneath the plant, we thought at the beginning of the season for the same reason, is it, I'm not a farmer.
I don't know about wheat straw in particular, unfortunately,
Okay.
but farmers do a lot of things.
Yeah.
Okay, any other question?
Can that be used for fruit or flowering plants, or is it just green?
So these plants are actually corn plants, and we chose to use corn plants. They're a very sturdy plant, but they show signs of nutrient deficiency almost immediately. So you see purple almost, actually you can see it there, if I want to slower this
Wait a
one
second,
on
wait a
the
second.
distilled water control. So it was easier to quantify the results and be able to get what we needed out of it. But yes, you could use it on anything. It's completely safe. The silica is sand, and there's such a small amount of it that it dissolves into essentially nothing. And the alginate is found in smoothies that we drink, so there's no harm in putting it to a plant where you're going to eat the fruit or vegetables that it creates.
So you can also see here, this is only grown for 2 to 3 weeks, and you can already see some nitrogen deficiency in these two. See how the leaves are not as green as the other ones? They're lacking color in the middle. So yeah, so they show it very quickly, which is why we picked it, so that we can get quicker results.
But yes, very safe.
What happened to the plant?
Sorry?
Pardon?
What happened to those plants now?
Oh, those plants that in the picture?
Yeah.
Yeah, well, we've had to harvest them, so they're.
Just grow it out, I want to see how many corn they can come out.
Oh, actually,
That would be really good
I
compared
didn't
to.
have a
Yeah,
photo.
it's one
We
thing.
got a baby corn.
Yeah,
Oh, really?
in the
One of
1
the
month
Evergrow
period.
ones. I
You're
didn't.
very
I
happy.
forgot. I totally forgot that photo.
Yeah, but we had to
Yeah.
uproot them to see the weights and be able to quantify all of our
Yeah.
results. We couldn't let them all grow out, but
Yeah.
I mean, again, future work, we can definitely let the plants actually live instead of
I think the measure
uprooting
of
them.
yield is more important than how the height is these days, really. I think it's one job.
Yeah.
Yeah.
You're right there.
Okay,
Anything else?
thanks speakers, and we'll move on.